So the next part of this session would be on helicases, led by Tim, Matthew, and Matthew from READDI Consortium, another AViDD consortium. They'll be telling us about helicases and drugging them. And after that, we would open up the floor for panel discussion around these two talks. So over to you guys. Tim, I think you're on mute.
There we go. Thanks, Alpha. So I'm going to give a quick introduction. I'm going to pose this question about whether helicases are a druggable protein family. We're very interested in this fascinating family of motor enzymes, and want to ask this specific question whether they will be druggable in terms of developing compounds that will be acting antivirals. So let's just change my pointer here. So I'm going to give a quick introduction as background, and then I'm going to hand it over to Matthew and Matt, who will give a little bit more details about some of the ways we're trying to answer this question. So helicases are this family of motor enzymes. They have a cofactor of ATP, which they hydrolyze to ADP, and they unwind double-stranded RNA or DNA in some cases, coupled to this ATP hydrolysis, and they can also process one nucleotide at a time along the RNA or DNA. So if we're thinking about how to drug this family of motor enzymes, there's some ways we could think about that. One would be to compete with the RNA, where it binds into the enzyme. You could also potentially compete with the ATP cofactor. Finally, you could think about molecules that would actually just jam up the motor, so binding to potentially allosteric sites that stop the movement of the enzyme. So these helicases are widely distributed across species. You can find them not only in viruses, but you find them in bacteria and in eukaryotes. They've been classified into 6 broad families, of which the SF1 and SF2 families is where the vast majority of the helicase reside. And you can find examples of RNA helicases in both the SF1 and the SF2 families. The characteristic of the helicases is that they have this helicase core, which is composed of 2 REC-A domains. And these cores have, even if the sequence identity is low, they have a conserved fold, which allows you to recognize these helicases belonging either to the SF1 or to the SF2 family. If I can skip this, there we go. So the SGCs had a long-standing interest in helicases as a family as a protein family. If you go into the PDB, there are more than 20 structures deposited by the SGC of REC-A-like helicase structures. And Matthew in Toronto has actually now built a database where he has compiled all of the PDB structures. This is in this ChemBioport of starting initially with all the human helicases. So there's more than 100 human helicase structures in the PDB. He's compiled all these into this database and shown the relationship in terms of sequence. And then this database has also annotated in there all of the structures where there is a small molecule bound in the structure. And so if you go in this database, you can mouse over this wheel here, and it pulls up all of the structures that are found in the PDB bound to that particular helicase. In most cases, the molecules that are bound are ATP, ADP, or ADP analogs. But there are a few examples where there are actually small molecules in these structures. So that's the first evidence that at least helicases of the family are ligandable. Matthew is now expanding this analysis to include the helicases in all of the viral pathogens of interest. And so that'll be one place to where we can find this information and people can look at the structures of compounds that bind to these helicase enzymes. The other way we're answering this question is through looking in public databases like Chembl. So this is the work of Holly Jo Martin at UNC. She is compiling a database of by analyzing Chembl for every entry where there's a small molecule which is annotated with activity on a helicase. So she found initially more than 20,000 entries, and then by a process of curation and annotation, she has cleaned up this data, so she now has about 13,000 entries where there's a clear annotation of activity against a putative helicase enzyme. And so she's going to continue to refine this, analyze it by whether they're DNA, RNA helicases by organism, whether they belong to the SF1 or SF2 subfamilies, and wherever possible, identify where the binding site of those molecules are. And again, the goal there is to have this annotated database available for people to look at it and decide if some of those molecules are starting points for developing inhibitors of viral helicases. And then, of course, you can just go into the chemical literature and look for examples of helicase inhibitors where people have developed small molecule drugs. So there is some evidence, I would say limited evidence of druggability. Molecules have been identified either by phenotypic screening or in biochemical assays. Probably the best examples are inhibitors of the helicase primase that's in either herpes or zoster. There's an example, amenovir, which has been approved in Japan as a treatment for shingles. This is inhibitor of the zoster helicase primase. And then pratelivir is a very interesting helicase primase inhibitor. It works on a cyclovir-resistant HSV. So this is HSV that's developed resistance to inhibition of the polymerase. And now you can go in with a helicase inhibitor and you can inhibit this acyclovir-resistant HSV. So that talks about some of the promise of helicase inhibitors as direct-acting antivirals. Now, there's no structure of how these helicase inhibitors bind to their target, but from mutagenesis data, it's known that they bind somewhere in the interface between the helicase and the primase. There are some nice structures of small molecules bound to some human DNA and RNA helicases. For example, this molecule that is 2 is an inhibitor of a BLM helicase, and its structure shows that it binds on a non-canonical site or allosteric site that is on the other side of the ATP pocket, somewhere in the vicinity of where the DNA binds. And then this structure of this BRR2 helicase is an RNA helicase. This is work by the group at Takeda. They found 2 different series of inhibitors. One, which binds again on a pocket that's distinct from the ATP site, somewhere close to the RNA site. And then another series of compounds that binds in the interface between the REC-A and the 2 REC-A domains. So at least from the search of the chemical literature, there is evidence of some limited druggability, but interestingly, the molecules that have been successful do not bind to the ATP or the RNA DNA sites, but appear to bind to allosteric or non-canonical sites. So they appear to be jamming up the motor rather than competing with the substrates and cofactors. And then, of course, there is the work from Joe Newman and Frank von Delft, who have done a fragment screen of the SARS-CoV-2 NSP13, and they placed all of that data in Fragalysis. So they have found more than 50 compounds which bind to various sites on the surface of NSP13. These are probably all millimolar binders, but provide potential chemical starting points for development of inhibitors. So we have a number of questions then in thinking about the druggability, specifically of SARS-CoV-2 NSP13. Ones which we would invite anyone to give input or ideas in terms of trying to solve this, because I think trying to answer these questions will help the whole field in terms of developing drugs against SARS-CoV-2. The first is, if you're going to screen the helicase, what's the best approach? Do you just do a full diversity screen, or do you use knowledge of binding sites to do targeted screening? And then what assay should you use to run the screen? Should you run an ATPase assay, or should you run a helicase unwinding assay, or should you just run a binding biophysical assay to screen? And then in all cases, what should you use as a positive control? I mean, we're not aware of a confirmed robust micromolar NSP13 inhibitor. So as far as we know, there's no positive control for any of these assays. With the fragment structure, what are the best approaches for going from millimolar to micromolar in terms of fragment elaboration? And Matt will talk a little bit about that challenge. And then, of course, there's a number of virtual docking approaches that can be used. There are multiple structures of NSP13. If you're going to do virtual docking, should you target the ATP RNA or one of the non-canonical sites? And Matt, you'll talk about cache challenge number 2 and how multiple labs are going to try and address this competitively to find the best approach. And so, yes, we're very happy to present here at the AViD Open Science Forum, because our goal is to be able to access and use all available ideas and share all results in real time to try and solve this fundamental problem of whether these helicases can be druggable and useful as targets for direct-acting antivirals.